Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Students, hope so you all are good, fine, and healthy. Well, in our previous section, we have discussed about the meiosis 1 in detail. Now we are going to move to discuss about the meiosis 2. So let's see what happens there. Okay, students, in meiosis 1, we have learned about that the interphase used to happen, and this interphase is totally similar to the interphase of the mitosis. That is like that in the interphase, that is like G1 phase, then S phase, and the G2 phase. After that, the prophase used to happen. Now, what basically happens in the prophase? The most important event that is known as a crossing over, if you remember that we have learned in detail. So in prophase 1, the crossing over used to take place, and after this crossing over, the chromosomes which are in the bivalent, you can say, or the tetrad bivalent condition, they used to arrange themselves in the center of the cell, and they used to in the equator, like they are going to arrange themselves in the equator of the cell, and that will be known as the metaphase plate. You know what all about the spindle fibers, which we have already discussed about, that those fibers who are going to attach with the protein will be known as a kinetical fiber, and those spindle fibers who are going to attach with each other from the opposite pole will be known as a non-kinetical fiber. Now, after the metaphase, there is an anaphase. So, what happens in the anaphase? Basically, in the anaphase, you can know that you know very well that these kinetical fibers are going to pull, they are going to get shorter, and they are going to pull the homologous chromosome like toward, you can say that a chromosome toward itself. Now, after the anaphase, the telophase used to occur, which is the reverse of the prophase. And in the prophase, you know very well that the nucleus is going to reappear, and this in the telophase, and obviously this chromosome is again going to convert into chromatin, and the nucleolus is going to reappear, and the telophase, obviously with side by side the cleavage will also occur, and that will result in the cytokinesis. Now after that, now what we are going to learn? After that, we are going to learn about meiosis 2. So basically, students, the most important thing which we should know before going on is that, listen to me very carefully. In the meiosis 1, what happens? A one parent cell used to split into two daughter cells, and that is 23 or 23. This means that the daughter cell which is produced having the half number of the chromosome as compared to the mother cell. Yes, these chromosomes were half, and that was like 23. But if you see the condition of the chromosome, then they are in this form. So you know very well, I have told you before, in that condition the chromosome are going to be 23, but what will be the number of chromatin? Obviously the number of chromatin will be the 46. So before going to the meiosis 2, you should keep this thing in your mind, that in the meiosis 1, a parent cell is going to divide into two daughter cells, and these two daughter cells are having the half number of the chromosomes as compared to the parental cell, but having the 46 numbers of the chromatin. So let's move to the meiosis 2. So students, in the meiosis 2, after the meiosis 1, the meiosis 2 is going to begin. In the meiosis 2, obviously the interphase will also be there, and that will be known as the interphase 2, or it also will be known as the interkinesis. Now students, listen to me very carefully. This interphase is quite different as compared to the interphase 1. Because in interphase 2, what is happening? There's a G1 phase and G2 phase. There is no S phase going to occur in the interphase 2. So you know very well, in the G1 and the G2 phase, the cell used to basically grow in the size and used to prepare themselves for the duplication, and used to increase the number of the enzymes, proteins, and used to double the organelles. But in the S phase, you know very well that the duplication of the chromosomes used to occur. No, when there is no, you can say, when there is a no S phase, so obviously there's no duplication of the chromosome is going to occur. This means that if the parental cell is of having the 23 of the chromosomes, yes, it will remain 26 number of the 23. If a parent cell have the 23 number of the chromosome, then it will remain 23 number of the chromosome. There is no duplication. It means that the parental chromosome will remain 23, and so there is no duplication, there is no S phase. So this is basically the difference in the interphase 2. Now what? Rest of the whole meiosis 2 is similar to the mitosis. And we have discussed the mitosis in detail, so I'm just going to give you a little review. Like obviously in the prophase 2, that is what is going to happen. You know it very well. The nuclear membrane is going to degenerate and the nucleolus is going to degenerate, and this whatever that you can say the thin-like structure, the thread-like structure chromatin is going to condense itself into the chromosomes, and as well as the centrioles are going to duplicate their self and used to move to the opposite pole, you can say. So this membrane is going to degenerate and the nucleus is going to degenerate, and the chromosomes are going to arrange their self into the center of the cell, you can say. They are going to spread their self all around in the cell, and now the spindle fibers are going to produce, and these spindle fibers are going to attach the canonical protein of the chromosomes from the both poles. So this was basically in the prophase. And after the prophase, what is going to happen? After the prophase, the anaphase 2 is going to begin. In the anaphase 2, you know it very well that metaphase, that is like after the prophase, that is the metaphase. So after the metaphase 2, what is going to happen? These chromosomes are going to arrange themselves in the center of the chromosomes and are going to make the metaphase plate, whereas the spindle fibers are going to attach from the centrioles. The spindle fibers are going to produce and they are going to attach the chromosomes, you can say, from each side. Are you listening me, students? It is like that each you can say the canonical protein is going to bond with the spindle fiber from the both sides to each centriole. So they are going to bond the canonical protein. The spindle fibers are going to attach with the canonical protein, and there will be obviously the non-canonical proteins. And after that, students, obviously there will be the anaphase, and in anaphase, you know it very well. In the anaphase 2, these you can say spindle fibers are going to get shorter and they are going to pull. Yes, they are going to pull the chromosome toward themselves. And in telophase, then in the next is the telophase. In the telophase, obviously that is the reverse of the prophase. The nucleus is going to you can say the nucleus is going to appear again, and the nucleolus is going to appear again, and side by side there will be the cytokinesis will also occur. Now what is basically the result here, students? The result basically is that these two parent cells having the 23 number of the chromosomes after the meiosis 2 is going to form the 23 number of the chromosomes. You know very well why I'm saying that the mitosis is basically the meiosis 2 is similar to the mitosis. The reason is that you have learned in the mitosis, I have told you that the meiosis 2 is similar to the mitosis. The reason behind is that in mitosis, the parental cells are basically as it, you can say that they are the editor copy of each other. We have learned in the mitosis that in mitosis the parental cells used to produce the two daughter cells, and which are totally resembling to the parental cell and having the same number of the chromosome that is present in the parent cell. That's why I said that the meiosis 2 is totally similar to the mitosis. So students, here we have where the two cells are going to produce the four cells and having the 23 number of the chromosomes. In the start, the chromosome were like having the sister chromatin in that case. Now in the daughter cell, the chromosome having the one chromatin. So this is it, basically this is the meiosis 2. So students, in this diagram, we are basically just going to go through that what is basically happening. So in the prophase 2, what happens that is totally similar to that the nuclear membrane is going to you can say degenerate, and after degenerating of the nucleus membrane, the nucleus is there, which is also going to disappear, and the chromatin, a thread-like structure of a chromosome, is going to convert itself into the condensed structure of the chromosomes. And you can see here that this is a cell 1 and this is a cell 2, which is basically produced, you can say that, which is basically produced from the meiosis 1. You know that meiosis 1 have produced two cells, and these here we have two cells, this one and this other, and they are going to further subdivide. So in metaphase 2, you have seen that they all have arranged themselves in the equator of the cells, and the spindle fibers are there. The kinetical fibers are going to attach with the kinetical protein of the chromosomes, and the non-kinetical fibers are going to attach with each other. Now in the anaphase, you have seen that the spindle fibers are going to get shorter themselves, and they are going to pull their chromatin toward themselves. And after that, there is an telophase. Telophase is the reverse of the prophase. The nuclear membrane is going to reappear, and the nucleolus is also going to reappear there, and the spindle fibers are going to disappear, as well as now each cell with the side by side the cleavage will also occur with the help of the cleavage Führer, and one cell is going to split into two cells, and each cell will have the one central. Yes, exactly, one centrosome. So, well, if you are going to see here, the same scenario is going on, but the end result is that these two cells are going to produce the four cells, 1, 2, 3, 4, and these fours having the 23 number of the chromosome, like each cell having the 22, 23, 23, and 23 number of the chromosome. But what is the difference there, that in the parental cell the chromosome were having the two chromatin, sister chromatin, but in the daughter cell there is a one sister chromatin, but the number of the chromosome is same. So that's why I've said you that in the start the meiosis 2 is similar to the mitosis 1, because the parents are totally producing the similar copies of the daughter cell having the same number of the chromosome. Hope so you get that. Well, but the main thing is that one of my students have asked a very good question. She asked for me that if a cell having 23 number of the chromosome, then obviously this cell will be the abnormal, because you told us before that the human cells are going to have a 46 number of the chromosome. Well, listen to me very carefully, student. Reproduction. Well, one of the greatest significance of the meiosis is the reproduction. Basically, the meiosis is occurring in the reproduction. Meiosis is basically occurring in reproduction. It is actually helping in the reproduction. Without meiosis, obviously, that is impossible that human cells are going to maintain their chromosomes in them. So, by the help of the meiosis, the human actually germline have maintained the number of the chromosomes in the cell. Now, what actually I mean, I'm going to explain you. Listen to me very carefully. Basically, better, we have learned about the reproduction, that there are two types of the reproduction. The one is the asexual reproduction, and other one is the sexual reproduction. Well, in the asexual reproduction, only one parent is involved, while in the sexual reproduction, the two parents are going to involve, like just like the animals, like most common animals, or like human. So what is going on? Basically, the sexual reproduction is going on. Now, how meiosis is actually helping in the reproduction? Basically, beta, you know that as I told you before, that in sexual reproduction the two parents are involved. One is male and other one is a female. So what is happening? A male having the 23 number of the chromosome, or 23 number of the chromosome cell, and the female having 23 number of the chromosome cells, these both cells are going to join and are going to form the 46 number of the chromosome. Are you getting my point? So this 46 number of the chromosome is going to form the zygote, and from the zygote there will be a complete baby. So what you get by that, students? Basically, this from where this 23 number of the cell come, like how a body having a 23 number of the chromosome in the cell. So obviously this is by help of meiosis. Yes, so meiosis is basically helping to produce a cell having the 23 number of the chromosome, and this is only this kind of the thing is better only happening in the reproductive part of the body. Like it is only in the reproductive part of the body. While a chick is producing a egg, like, and from that egg the whole baby is going to develop. So basically this egg have a 46, like you can say, sorry, it's not like chicken have different chromosomes. I should not have discussed this example. Okay, let's discuss something in detail. Basically students, if let's suppose if a father is going to give the 46 number of the chromosomes, and a female is also going to give the cell having the 46 number of the chromosome, and they are going to unite and they are going to produce how many, like 12, 6, 6, 12, and 1 carry 8 and 92. Like it's mean a human having the 46 number of the chromosome is going to produce a baby having the 92 number of the chromosomes, and obviously that baby will not be the human, he will be something like alien. So if let's suppose if this baby is going to be someday father, okay, so this father is going to produce the 96 number of the chromosomes, and let's suppose a mother is all going to produce also 92 number of the chromosomes, and obviously this time the baby is going to produce will be, my god, that is a 4 and 184 chromosome. Oh, this is so ridiculous. So better basically to maintain this, the Allah Ta'ala have created a phenomena that is a meiosis. Basically this meiosis used to do the half, you used to create a cell having the half number of the chromosome. So what is the advantage of the having the half number of the chromosomes? Let's suppose by the help of the meiosis the father have 23 number of the chromosomes in a cell, and mother have also the 23 number of the chromosomes, and now when they are going to form a egg or zygote, when they are going to unite with each other, they are going to form the zygote, and now this zygote will have a 46 number of the chromosome, and yes, this 46 number of chromosome is going to produce a normal baby. Yes, so basically the one of the greatest significance of the meiosis is to maintain the germline. Now this 46 number of the chromosomes are when this baby someday will be a father. So obviously this is also going to, by the help of the meiosis, again the half number of the chromosomes will be there, and from the mother half, and then again the 46 number of the baby is going to produce. So normal babies are going to produce by this act. So beta basically meiosis is actually rest of the body there's a mitosis is going on. Only in the reproduction, in the reproductive area, only the meiosis is going on, and meiosis is too much necessary at that part. The reason is that the meiosis is going to maintain the germline, like we are normal. The reason is that the meiosis is there. So hope so you get that, and if you have any query you can discuss with me, you can command me. I will try my best to resolve a query, otherwise we will discuss directly. Jazakallah khair. Assalamu alaikum wa rahmatullahi wa barakatuhu.